Welcome to this module on the digestive system. During this module, you will learn about the different components of the digestive system and how the nutrients in our food are broken down and absorbed by the body. The digestive system is the body's own food processing factory. It is responsible for breaking down and absorbing nutrients. The overall function of the digestive tract is to process ingested foods into smaller molecular forms that can be transferred, along with salts and water, from the external environment to the internal environment of the body. Digestion and absorption of all nutrients takes place within the gastrointestinal tract. The structure of the gastrointestinal system includes the gastrointestinal tract and the accessory glands, which are the salivary, the pancreas, and the liver. The organs that make up the gastrointestinal tract are the mouth, the pharynx, the esophagus, the stomach, the small and large intestines, and the anus. Each of these components has a precise function and is involved in breaking down different food components. In the mouth, the enzyme salivary amylase starts to break down carbohydrates. In the stomach, the enzyme pepsin breaks down proteins, and in the small intestine, there are the enzymes lipase, pancreatic amylase, and trypsin. Lipase breaks down fats, pancreatic amylase breaks down carbohydrates, and trypsin breaks down proteins. In order for food to move through the system, it goes through a process called peristalsis. The movement pushes both liquids and foods through the gastrointestinal tract and mixes contents within each of these organs. The way that the digestive system works is through these enzymes that help to break down the large molecules into smaller ones. This means that when carbohydrates are ingested, they are broken down into glucose, fats into free fatty acids and glycerol, and proteins into amino acids. Our gastrointestinal tract receives innervations from neurons of the autonomic nervous system, which is both sympathetic and parasympathetic, which constitutes the extrinsic innervation. This means that digestion is an autonomous process. To understand fully the complexity of the digestive system, we will explore the functions of each of the components. The oral cavity, which is also known as the mouth, is the first portion of the gastrointestinal tract. This is where mastication, or commonly known as chewing, of food starts and begins to break down the food. Our saliva moistens our food and also contains the enzyme amylase, which helps to break down larger carbohydrate molecules. The next structure is the pharynx, which is the opening of both the digestive and the respiratory system. Next we have the esophagus, which is around 25 centimeters long and extends between the pharynx and the stomach. The esophagus has two sphincters, the upper esophageal sphincter between the pharynx and the esophagus, and the lower esophageal sphincter between the esophagus and the stomach, which is near the cardiac opening. Both sphincters regulate the movement of the ingested meal into and out of the esophagus. The action of peristalsis helps to move food down to the stomach. The stomach is an enlarged part of the gastrointestinal tract in the upper portion of the abdomen. The lower opening of the stomach communicates with the proximal segment of the small intestine, which is also known as the duodenal bulb. Gastric juices contain acids and enzymes which help to break down food. Hydrochloric acid kills any bacteria, whilst pepsin breaks down proteins into shorter peptides. The small intestine is a long tube which consists of three portions: the duodenum, the jejunum, and the ileum. This is the main site of digestion and absorption, as the duodenum villi absorb nutrients into the bloodstream. The large intestine includes the cecum, the colon, the rectum, and the anal canal. Within the large intestine are bacteria that can help to produce some vitamins and fight infection. The gallbladder is a long pear-shaped organ found under the liver. It functions to store bile, a compound produced by the liver to digest fat and help the body absorb fat-soluble vitamins and nutrients. Now let's move on to the digestion and absorption of the different macronutrients. We'll start off with carbohydrates. The goal of carbohydrate digestion is to break down all disaccharides and complex carbohydrates into monosaccharides, which are single molecules of sugar. The only exception here is fiber, which is not completely absorbed by the body. Some examples of disaccharides are lactose, sucrose, and maltose. And some examples of complex carbohydrates are starch, glycogen, glycolipids, and fiber. Digestion begins in the mouth when salivary A amylase is released when we start chewing. This sends a signal to the gastrointestinal tract that carbohydrates are being consumed and other secretory enzymes are released further down. Amylase breaks the starches into maltose and polysaccharides, however only 5% of starch is broken down here due to limited exposure. Once the carbohydrate gets to the stomach, minimal digestion occurs here. Instead, the acid environment is there to kill bacteria, as amylase is inhibited in an acidic environment. Once the carbohydrate reaches the small intestine, pancreatic A amylase is released, which breaks the molecule down to simple components. Further digestion of oligosaccharides, which are 3 to 10 simple sugars linked together, is done through enzymes on the brush border of the epithelium of the duodenum and jejunum. Major brush border enzymes are lactase, sucrase, A-dextranase, and glucoamylase. Once digested, the end products become glucose, galactose, and fructose. Carbohydrates not digested in the small intestine pass into the large intestine, where they are digested by colonic bacteria. This results in the release of short-chain fatty acids, propionate, butyrate, and acetate, along with methane. The short-chain fatty acids provide vital nutrition to colonocytes, which are the healthy bacteria in our gut, but excess volumes induce diarrhea and abdominal cramping. The process of absorbing carbohydrates is extremely complex. There are several mechanisms which transport end products of digestion across the intestinal mucosa and into the blood, which involve sodium channels. Digestion and absorption are typically coupled with the enzymes being closely located with the appropriate transporters. Glucose absorption occurs in the small intestine by active transport via the sodium-glucose cotransporter. Galactose, fructose, and some glucose absorption is completed by the GLUT5 transporter by facilitated diffusion. An example of this process is shown in the image here. The glycemic index, or for short GI, is an indicator of how rapidly blood glucose levels rise following a carbohydrate-containing food. Foods that have a low GI do not raise blood glucose levels as fast as foods that have a higher GI. A diet of lower GI foods has been shown in epidemiological and clinical trials to increase weight loss and to reduce the risk of obesity, type 2 diabetes, and cardiovascular disease. Examples of foods and their GI content is shown in this picture here. The key points from this section on the digestion and absorption of carbohydrates is digestion starts in the mouth and finishes with the absorption in the large intestine. Different enzymes present within the gastrointestinal tract function to break down the various carbs in the diet. The end products are glucose, galactose, and fructose. Colonic bacteria digest carbohydrates, which aren't broken down further up in the gastrointestinal tract. And colonic digestion creates short-chain fatty acids, which provides us with energy. Next, we have the digestion and absorption of fats. Lipids, or otherwise known as fats, are large molecules and are generally not water-soluble. Unlike carbohydrates and proteins, they must be broken down so that we can absorb them. Remember, fats are made from glycerol backbone and 3 fatty acids. The breakdown of fat is by far the most complex digestion process because there are vital nutrients which are insoluble in water that have to transfer from one aqueous environment to another through cells. This process is solved by emulsification and specialized enzymes. The process of digesting fats begins in the mouth. Chewing coupled with the action of emulsifiers enables the digestive enzyme linguinol lipase to begin digestion. As a result, the fats become tiny droplets and separate from water components. Gastric lipase within the stomach breaks down the fats further before entering the small intestine. As fats are largely insoluble in water, the digestive system has to counteract this as it needs to combine the fats with internal water. This is done through bile. Bile contains bile salts, lecithin, and cholesterol and is made in the gallbladder. Bile works to hold onto the fat molecule while simultaneously holding onto water. This process of emulsification increases the surface area of the fat, which makes it more accessible to our digestive enzymes, known as lipolytic enzymes. Once pancreatic lipase enters the small intestine, it breaks down the fats into free fatty acids and monoglycerides. Bile salts then envelop the short and medium chain fatty acids and monoglycerides to form micelles, which have a water-soluble exterior which allows them to be transported to the intestinal microvilli. This is how a non-water-soluble compound becomes water-soluble for digestion. The same applies to transport through the blood for absorption. Inside our intestinal cells, the broken down monoglycerides and fatty acids reassemble to form triacylglycerols. Triacylglycerols, cholesterol, and phospholipids join to become lipoproteins, which can then form a chylomicron able to be released into the bloodstream through the lymphatic system. The key points from this section on the digestion and absorption of fats are: the process of digesting fats is through B-oxidation and requires the use of oxygen through an aerobic pathway. In the small intestines, bile emulsifies the fats, which enzymes then digest. Intestinal cells absorb fats. Long-chain fatty acids form a lipoprotein structure called a chylomicron. The chylomicron transports lipids into circulation. Short and medium chain fatty acids can be absorbed directly into the bloodstream because they are water soluble. Fat is used as energy when energy supplies are low through a process called B-oxidation. Fats can be stored as energy, known as triacylglycerols and triglycerides. Fatty acids can be unsaturated or saturated. This brings us on to the digestion and absorption of proteins. Firstly, we will revisit the structure of protein, as it is important in the understanding of how proteins are digested. Proteins are sequences of amino acids linked together by peptide bonds called polypeptides. Proteins must be digested into amino acids or di- or tripeptides. The process of digestion of dietary proteins begins in the stomach. In the gastric lumen, pepsinogen is secreted from chief cells and hydrochloric acid from parietal cells. Hydrochloric acid denatures the proteins and also converts inactive pepsinogen to active pepsin, which hydrolyzes the proteins to amino acids and smaller peptides. In the duodenum and small intestine, the proteases secreted by the pancreas play a role in protein digestion. The proteases are trypsin, chymotrypsin, and carboxypeptidase. Up to 70% of proteins are converted to oligopeptides here through the actions of the pancreatic proteases. Oligopeptides have a more rapid absorption than free amino acids. Additional digestion occurs at the brush border, which increases the amount of protein for intracellular transport. The principal products of protein digestion are small peptides and amino acids. These free amino acids are transported by several mechanisms in the duodenum and the jejunum. Some amino acids share the same transport system, so if there is a large amount of one particular amino acid, this means that the absorption of others may be inhibited. The key points from this section on the digestion and absorption of protein is pepsinogen is secreted, which is then converted to pepsin by hydrochloric acid. Hydrochloric acid hydrolyzes the proteins to amino acids. Digestion occurs at the brush border, which increases the amount of protein for intracellular transport. And free amino acids are transported by facilitated diffusion to the blood vessel. We now just want to briefly touch on the function of the pancreatic juice in the digestive system. The pancreatic juice is extremely important as it contains enzymes for digesting carbohydrates, proteins, fats, and nucleic acids. The major pancreatic proteolytic enzymes are trypsin, chymotrypsin, elastase, and carboxypeptidase, which we have touched on during this module. The non-proteolytic enzymes are amylase, which is a carbohydrate digesting enzyme which degrades starch into oligosaccharides, ribonuclease and deoxyribonuclease, which are nucleic acid digesting enzymes, and lipase, which degrades triacylglycerols into fatty acids and glycerols. This module has gone over the digestive system and the process of digestion and absorption of each of the macronutrients in quite a lot of detail. You may not be required to give advice to your client that is directly related to the content of this module, but it is vital that you have a good understanding of all the different systems in the body and all the different processes.